You're at the cutting edge of cardiovascular sciences. I'm your host, John Cooke. I'm professor and chair of the Department of Cardiovascular Sciences here at Houston Methodist Research Institute. And we have a really great show for you today on metabolic memory and how it affects cardiovascular complications. Joining me in the studio is Dr. Li Lai. Dr. Lai is one of my junior faculty. She received her PhD training in China, Shanghai, China, at East Normal University before she came to Texas A&M as a scholar there, and then joined my laboratory in 2014, where she's done some really exciting work on angiogenesis and how metabolism influences angiogenesis. Joining us as our guest today, Dr. Rama Natarajan. Thank you for coming, Dr. Rama Natarajan. Rama is professor and chair at the Department of Diabetes Complications and Metabolism within the Diabetes and Metabolism Research Institute at the City of Hope National Medical Center. She was the chair of the Women's Leadership Committee at the ATVB Council of American Heart Association, and there she promotes the advancement of women in science, particularly vascular sciences. She holds the National Business Products and Industry Endowed Professorship in Diabetes Research. Dr. Natarajan is a leader in the field of diabetes-related vascular complications and a pioneer in diabetes epigenetics. Her work has provided clinical and epigenetic evidence for metabolic memory in cardiovascular complications. She, in particular, she's shown that prior episodes of high blood sugar can predispose diabetic patients to long-term vascular complications. She's published over 200 papers. She has numerous awards. She's been continuously funded by the NIH for 25 years, and it's a great pleasure to have you on today, Rama. Thank you for joining us.
Thank you, John. Can you hear me?
Yeah, we can hear you great.
Okay.
You're at the City of Hope now, speaking from the City of Hope about your work in diabetes and vascular complications. Thank you for joining us, and please begin to share your presentation.
Sure, right. I'd like to thank, can you see my slides now?
You can see your slides great.
Sounds good. Okay, sounds good. But slides are not moving. Okay, let me try again before I start talk. Okay.
You can see it.
Okay. Can you see it?
Yes, looks good.
Okay. Okay. Thanks a lot, John, for the great introduction. It's indeed a pleasure to participate in this important educational event. And, I've been following the wonderful work going on at Houston Methodist, and it's great to be part of this Vascular Center series that you have. So as John was saying, well, in my lab, we've been working for numerous years on the complications of diabetes, particularly cardiovascular complications. And we know that diabetes has increasing prevalence, but the diabetes itself has quite a few drugs available. But the main problem with diabetes is the fourfold increased rates of complications, cardiovascular complications like hypertension and atherosclerosis, and microvascular complications like diabetic nephropathy. And there's a strong connection between renal and cardiovascular complications. And it's also been shown that diabetic complications seem to be progressing despite glycemic control. And this is very frustrating for many patients. And clearly, genetics alone cannot explain this because GWAS has found very few candidate genes for diabetic complications. So for a number of years, our lab has been focusing on looking at the role of epigenetics, non-coding RNAs. So just to quickly illustrate the havoc that diabetes does to the vascular system, to the cardiac and the vascular system. So many, many years back, we collaborated with Ross Gerety, a very famous vascular pathologist, and he had this swine model of diabetes-induced accelerated atherosclerosis. So on the upper panel, you see the left anterior descending artery of a pig that is both hyperlipidemic as well as diabetic. At the bottom, this pig is just hyperlipidemic. And clearly there's a dramatic difference. The diabetes pig has over 95% stenosis and severe calcification, all features of human diabetes-induced accelerated atherosclerosis. And what we found was there were greater indices of both inflammation and oxidant stress in monocytes from these diabetic and hyperlipidemic pigs. So this set us off, and we and others found that inflammation and fibrosis are major culprits in most diabetic vascular complications. And we looked at the molecular mechanism of regulation of these inflammatory and fibrotic genes regulated by the usual suspects like high glucose, advanced glycation end products, various growth factors. But how this is associated with accelerated atherosclerosis and nephropathy. So the next step is I started thinking about what's really happening at the level of chromatin. Is there a role for epigenetic mechanisms in diabetic complications? So why are we interested in epigenetics? This is because epigenetics, this is the layer on top of your genes and genetics, and it actually can control gene expression. It's altered by the environment and lifestyles, like how much exercise you do or you don't do. And chromatin is dynamic. It can exist in at least two states. Heterochromatin is the closed or condensed form, and euchromatin, which is the open form, easily accessible to the transcription machinery. The subunit of chromatin is the nucleosome, which is the octamer of histones, the N-terminal tails of which can be post-translationally modified by various modifications like acetylation, methylation. And these modifications can alter gene expression for more gene activation or gene repression, depending upon the location and type of modification. And whereas promoter DNA methylation generally associated with gene repression, and collectively the epigenome consists of DNA methylation, histone modification, etc. And this interfaces with non-coding RNAs. So a broad objectives of our lab was to look at epigenetic mechanisms, regulate the transcription of key pathological genes involved in diabetic conditions. It's involved both inflammatory, fibrotic, and of course, angiogenic genes too. The functional relevance of these chromatin changes and DNA methylation, non-coding RNAs, to increase inflammation, fibrosis, and in particular to metabolic memory, as John was alluding to, and I'll talk about it briefly. And then we are doing a lot of EWAS, that is epigenome-wide association studies, using techniques like ChIP-seq and DNA methylation profiling. And this is to look at epigenetic variations in disease states. And finally, I'll briefly talk about why this is important and how we can actually translate this to look at the translation potential. So metabolic memory was originally identified in the DCCT-ED clinical trial, which is the Diabetes Control and Complications trial, which was started around 1983 with about 1,500 type 1 diabetic patients. And what they asked a simple question: does strict glycemic control reduce the progression of microvascular and macrovascular complications? And what they found is at the end of less than 6 years, they had to stop the clinical trial ethically, because it was found that clearly patients in the intensive glycemic control are getting much lesser complications compared to those who were on conventional glycemic control. So at the end of about 7 years, they stopped the trial, but followed them in an epidemiological edict study where all the patients were placed on similar strict glycemic control and the hemoglobin A1Cs came together. But surprisingly, it was found that even to this day, which is beyond 30 years past the start of the DCCT trial, the patients on the original intensive control are getting much lesser complications. The right-hand panel shows the cardiovascular events. They're still much lower compared to the conventional treatment. And this has also been seen in type 2 diabetes, where it has been called legacy effect. So we asked the question, does epigenetics play a role in this? So we set up a simple, initially experimental model where we took vascular smooth muscle cells from these obese and tubby type 2 diabetic mice, which are called DB mice, and compared to the genetic control DB plus mice. And we found that these vascular smooth muscles, even though they were cultured for many passages outside the animal, they showed much greater migration, proliferation, kind of related to atherosclerosis in the diabetic cells compared to the non-diabetic cells, even after several passages. And so this is showing the increased expression of inflammatory genes. And conversely, we found that there was a decrease in this chromatin repressive histone modification, K9 trimethylation, at these inflammatory genes. That is, the loss of chromatin repression was associated with increased gene expression. And so we suggested that this persistent dysfunctional behavior of vascular cells and increased inflammation due to metabolic memory phenomenon, and this could be due to epigenetic modifications. So after a number of experiments,
may I ask you a question about that slide?
Absolutely.
If you could go back
to it.
I just thought that was very interesting that you have these two mice. One is diabetic and obese, and the other is not. And you're taking cells from these animals and then studying them in cell culture, and it appears that the cells from the diabetic mice are more proliferative. I think that's what you said. Is that correct?
Yeah.
And that was under the same conditions. They're in the same media, the same
exactly. Yeah.
Growth factors. So there's something going on here that the diabetes has done to these cells that has altered them. Interesting.
Right. Correct. Yeah. We think, and we have data. Actually, we're just doing a huge integrative omics study, and we have done DNA methylation. We've done a lot of, including ataxic, and we think that the chromatin is kind of locked in this persistent inflammatory state. The chromatin near these inflammatory genes actually becomes loosened. And whereas in the non-diabetic cells, they can come back because, we have to respond to an inflammatory stimuli like, coronavirus or any virus. And we do spew out inflammatory cytokines to kill those bacteria, but our chromatin automatically goes and closes back. What happens in diabetes is that they lose this capacity. This is what our data is showing. And we've shown this with other cell types. We have some data with endothelial cells also. And I think recently many people have done macrophages. So what we think is happening that under diabetic conditions, and this we also have data with hyperlipidemia, with various type of mechanisms, which I don't have time to talk about. We get a loss of these repressive marks from susceptible regions of the genome, leading to opening up of the chromatin. Increased activating marks are decorated on this part of the chromatin, and we get these pathological genes expressed. So, it's easy enough to do one gene at a time, but we wanted to graduate into more genomes. And so we are working on epigenetic roadmap to link epigenotype variations to diabetic complications and metabolic memory. And the hypothesis that hypoglycemia promotes epigenetic changes and the persistence leads to metabolic memory. So we have done EWAS, this is epigenome-wide association studies in various model systems, but I'll briefly touch upon our ongoing work with the DCCT-ED clinical trial. We've been very fortunate to collaborate with the executive committee of the clinical trial because they are very particular about how they share these precious samples from patients, because a study like DCCT can never be repeated. And but they were very generous with sharing the DNA samples, archived DNA samples. And so we have sets of DNA from a case group, which is from the former DCCT conventional treatment group, who are currently even beyond 30 years, still expressing greater complications, retinopathy, nephropathy, and macrovascular during EDIC, which is still ongoing. And a control group, which is from the intensive treatment group, who are not having progression of complications. And we have looked at both histone modifications as well as DNA methylation. So very briefly, in about the second or the third study, what we found that there were several differential methylated loci or DMLs, which show significant persistent changes in DNA methylation, that is memory. So we took samples from the same patient about 17 years apart, And we found that the same loci show exactly the same changes 17 years apart in the same patient. And one of these sites particularly caught our attention, and that is a gene called TXNP. Now, the 3' UTR has 3 CPGs, all of which showed consistent hypomethylation in 1993, and the same samples at 2010, both monocytes and lymphocytes from the same patient showed this consistent hypomethylation. And in this study, which is about 4 years old now, we showed a firmer connection to metabolic memory. And also we were interested in TXNP. And the fascinating part is that as soon as we published this, there have been a series of papers showing that exactly the same CPGs are also hypomethylated in type 2 diabetes, in dyslipidemia, insulin resistance. So something about this seems to have a memory of metabolic dysfunction. So we think it's a good target for complications and metabolic memory. And of course, it's well known to be associated with hyperglycemia. But this study, the Fourier study, had clearly supported an epigenetic explanation, but it was done with a small cohort, and there were other unanswered questions because we couldn't really make the clear association between hemoglobin A1C history and DNA methylation. And also, is there a clear association between DNA methylation and future risk of complications? So in order to address this, we undertook a very much bigger study with 500 randomly selected DNAs. And this study, which is a very big study, recently published in Nature Metabolism. So what we did is we've identified the A1C associated DNA methylation CPG sites. And we also very detailed investigation into the putative biological functions. And we also looked to see whether the CPG DNA methylation at these key sites, including the TXNP 3' UTR, are they associated with future complications development during EDIC? And we also examined for the first time that the DNA methylation actually has a mediation role in the well-known association between A1C and complications. So just about 2 slides from this recently published study. So this is a Manhattan plot showing the identification of this 186 CPGs from, collected from all these 500 patients. And we found that about 186 of them were associated with the A1C history. And we were fascinated to find that once again, TXNP stood out, and this time with much, much greater FDR and significance. And we were also able to do cross-cohort validations in other cohorts. And we showed that this association with A1C history persisted much later into EDIC.
So Rama, if I could ask you a question about that, Rama, that last slide, that makes a lot of sense to me that TXNP was the gene that was most deregulated or dysregulated in diabetes. That, of course, is an important protein involved in oxidant stress, correct?
Absolutely.
It inhibits,
Absolutely.
That inhibits, TXNP inhibits thioredoxifen, which is an antioxidant protein. So
correct. Yeah, that's the major mechanism. Yeah.
And oxidative stress is increased in diabetes, so it makes a lot of sense that this one is impaired.
Yes. So we are looking at the interesting part is that we also have data that this is dysregulated in bone marrow stem cells. So I'll just briefly talk about that, and then we can talk, discuss more.
And before you do, Rama, let me just remind our listeners that they can ask questions by text. All they have to do is they can text DEBAKEY, that's DEBAKEY, to 37607, and then text in the message. They can join that way, or they can join by web. They can go to pollev.com and enter DEBAKEY, and then respond that way. And I think that information is also somewhere in the, it's also displayed. Okay, Rama, please continue.
Great. And TXNP is also important because it's one of the major bad guys for the islet, the beta cell. Due to oxidant stress, TXNP has been shown to destroy beta cells. So it can actually be associated with diabetes itself, not alone just complications. So we used the chromatin states that have been defined by the NIH Roadmap Epigenetic Program to actually see where are these A1C-associated CpG sites, where are they clustering? And it was fascinating to see that a large number of these hemoglobin A1C-associated CpG sites in monocytes are enriched in enhancer regions, and whereas in the hematopoietic stem cells, they are enriched in transcription states. So suggesting that these differential methylated sites can actually have some functional consequence because they are in key regulatory regions. And then we also used the available DNase-1 hypersensitive site data from ENCODE, and we were again fascinated to see that these A1C-associated sites were highly enriched, or they were in the hypersensitive sites in stem cells and in myeloid progenitors, but not in lymphoid progenitors. And if we actually in vitro treat CD34 cells from humans with high glucose, we could see hypomethylation at the same 3 CpG sites. And there was also upregulation of TXNP. That's just one of the candidate genes. So from this recent study, we concluded, and so, yeah, there's a lot of other things that we did, but basically our data suggested that prior history of hypoglycemia may induce persistent DNA methylation changes at key loci, including TXNP and others. So DNA methylation can act as a mediator between A1C and complications. And interestingly, we think that the site of metabolic memory could be in stem cells because these DNA methylation are epigenetically maintained in differentiated myeloid cells, that is downstream. And this sustainment is likely through modifying enhancer activity at nearby genes. So we do a lot of systems biology of diabetic complications, and we use, all these techniques that are available, RNA-seq, ChIP-seq, ATAC-seq, Capture-High-C. We integrate all of these to gain new information about diabetic complications. And one of this has been very gratifying because we found a lot of very interesting non-coding RNAs, both microRNAs and long non-coding RNAs that have potential epigenetic action. So there's like a crosstalk between the non-coding RNA layer and the epigenetic layer. So we asked a question whether they promote fibrosis and inflammation. And this is important because most RNA transcripts are not protein coding gene, but actually non-protein coding. And link RNAs are particularly fascinating, and they keep me awake at night because they have diverse regulatory mechanisms, very complex. But what was interesting to us is they have epigenetic action. So we think that they can also interface with metabolic memory. And particularly recently, there's a lot of interest because they could be new biomarkers and therapeutic targets. So in particular, we have looked at whether there are link RNAs that can regulate inflammation in monocyte macrophages, because monocyte macrophage inflammation plays a role in insulin resistance, several diabetic complications, most diabetic vascular complications. So we've done a lot of profiling of link RNAs in monocytes and macrophages from both humans and mice with diabetes and atherosclerosis. So just to cut long story short, what we have found that a number of link RNAs, one of them is called MIST, another is called DNM3OS, another is called DRYER. And you can just check out our papers, recent papers. This one, the last one is just in press right now. So what we find is that by virtue of these RNAs binding to critical RNA binding proteins, they can actually drag these RNA binding proteins to the promoters or enhancers of their target genes. And this way change the chromatin around these susceptible genes. So apart from just histone modifications and DNA methylation, these link RNAs can actually even cause DNA architecturally folding up and bring promoter and enhancers together. And in this way, link RNAs can play a very important role in inflammation and vascular dysfunction. And recently, there have been link RNAs that have been identified in macrophages and vascular smooth muscle cells. And our lab, Gen's lab and other labs are also looking at endothelial cells. And these link RNAs are also in circulation, so they can be used as biomarkers. So apart from and interestingly, as my next couple of slides would show, so this brings me to my last couple of slides. So to the translational potential of epigenetics and link RNAs. And in this, I'll just talk to you about one study we did where we discovered that an endoplasmic reticulum stress-induced mega cluster of microRNAs, and which are hosted within a long non-coding RNA, we have shown that it actually plays a key role in regulating fibrosis in the early stages of diabetic nephropathy, both in vivo and in vitro. So we figured out a way to target this link RNA. I know many of you in your cardiovascular center are interested in RNA targeting. So what Dr. Kato in the lab, he designed a locked nucleic acid gapmer antisense oligo to target the first exon, the transcription of this long non-coding RNA, which is hosting all these 40 microRNAs. And what he was able to do is downregulate both the link RNA as well as several key microRNAs. And he was able to show that it actually protects diabetic mice, from key features of early diabetic kidney disease. We also have shown that it's effective in human cells. And so we are trying to translate this, bring it to human treatment ultimately. For this, we have created with CRISPR-Cas9 a humanized, partially humanized link megacluster mice. And we also use CRISPR-Cas9 to create 379 knockout mice, which is the first microRNA in this link RNA cluster. We also have made several link MGC knockout mice, and we are looking at the phenotype of these mice, which is showing some very exciting phenotypes. So to put it all together, we think epigenetics and non-coding RNAs are a critical layer in regulating diabetic cardiovascular complications and metabolic memory, whereas many of our labs have looked at several signal transduction mechanisms downstream of diabetes. But we think that this is also important to look closer into the nucleus at epigenetic changes, transcriptional regulation of non-coding RNAs, and how this vicious cycle among these epigenetics and non-coding RNAs can augment, accelerate, and amplify the vascular disease, and how that we actually need to find ways to interrupt this vicious cycle. And so basically, diabetes increases pathological genes via various epigenetic mechanisms. And because this, diabetes is a more open state of chromatin, which is primed for the transcription of pathological genes, and this crosstalk between epigenetic layers, many regions. So, and more important, we think that in the future, the cancer guys are already using epigenetic inhibitors for treatment. So we think it can be applicable to vascular disease. And as you know, the BRD4 inhibitors like JQ1, looking at super enhancer inhibitors, are already being investigated for cardiac failure, for cardiac hypertrophy, pulmonary hypertension. So there is a lot of opportunity for epigenetic therapy, because unlike genetics, you can't really change your parents, you can't change how you inherited your gene, but you can, epigenetics is reversible. You can choose how to live your life. So we have this very strong window of opportunity. So I'd like to thank all of my wonderful colleagues and collaborators over the years at City of Hope, including Art Triggs, John Rossi, and of course, Jen, Dustin, others, and our fantastic course at City of Hope, and the people in the lab, particularly Nancy, my bioinformatics specialist, who tries to help me to make sense of all the voluminous data we generate, graduate students and postdocs, and my collaborators at DCCT and Jocelyn, and of course funding, without which none of this can happen. Thank you very much, and I'll be happy to take more questions.
Rama, that was beautiful. Two things, it's so hopeful and so exciting. So exciting in many ways. I mean, this concept of metabolic memory is very cool. And I think for me, the most compelling data is from the clinical trials showing that 17 years after having been randomized to the group where you were less intensively treated, that had an impact 17 years later on the expression of genes that are important in cardiovascular disease, like the TXNP that you mentioned.
Right.
That's exciting and so promising, what you said at the end, that you can't change your parents, but you can change your epigenetics. So that's very exciting. We have a couple of questions from listeners. Are there any available techniques to knock out or modify methylation sites in order to establish more mechanistic descriptions about epigenetic phenomena? And I would add to that to maybe therapeutically reverse some of these changes.
Fantastic question. That's exactly what we're working on. And so there are ways to do site-specific alteration of DNA methylation. Can be done with, having VP64, where you can do site-specific activation of a promoter. So there are ways that Josh Tompkins, who's my collaborator, he has developed a technique where he uses CRISPR to actually focus on a specific CPG to either remove the DNA methylation or insert DNA methylation. So this is what we call epigenetic engineering or epigenetic editing. And I think, in fact, there was a recent paper on cancer stem cell. And also my collaborator, Josh, has been able to edit iPSCs and also cardiac stem cells and shown that this altered DNA methylation is carried over subsequent generations, almost 10 generations, into the downstream cardiac myocytes. So there are ways it can be done, and we are trying to do it in bone marrow stem cells right now.
Another question just popped up, and the question is related to these epigenetic changes that occur in diabetes. Have they been observed in cells other than vascular cells? You showed us nice data with vascular smooth muscle cells. What about other cell types?
Oh, yes, yeah. So we have shown it in kidney cells, and others have shown it in cardiac cells. So even in the kidney, we focused on, say, mesangial cells. People have looked at podocytes, they've looked at tubular cells, and there's a study supposed to be coming out where they've looked at macrophages, monocyte macrophages also. So, and Jen has a paper, we collaborated, and she shows it in endothelial cells, in high glucose and TNF-alpha treated endothelial cells. Yes, so yes, many, not only vascular cells, but other cells, many of them. And there are studies in retinal, so because retinopathy is one of the first complications seen in both type 1 and type 2 diabetes.
Do you want to ask the next question, Lily?
So next question is, does this epigenetic abnormality persist if we transfer cells into media or organisms without hyperglycemia? And does hypoglycemia induce epigenetic modification in a non-diabetic animal? So
can you quickly repeat that? I temporarily lost you.
So the question is, does this epigenetic abnormalities persist if we transfer cells into media or organisms without hypoglycemia? And
Okay, got it. Yeah. So people have actually done a bone marrow transplantation from a diabetic mice into a non-diabetic mice and vice versa. And yes, it does persist even after it's transferred from the whole animal itself. Yeah.
Interesting. So there's these epigenetic changes are to a certain extent locked in. They persist over multiple passages of cells, and even it sounds like through generations. You said you seem to indicate to us that some of the CRISPR-Cas studies with epigenetic engineering, those changes could persist through multiple generations. Is that correct?
Correct, through differentiated states. So in the case of some people who do transgenerational inheritance, they have looked at rats who are fed a high-fat diet, and they have data showing that even after multiple generations of the whole animal, not just cells. So they have shown that some of these epigenetic changes can be transmitted. And so these kind of studies are, depending upon the location of the epigenetic change, clearly it can be transmitted through generations in cell culture, as well as, during stem cell differentiation, as well as in the whole animal too. So the founder mice, and this is what they think is carried through the sperm, both non-coding RNA as well as DNA methylation is more stable, so it's a little bit easier to comprehend. But the histone modification transmission, there are data coming out that some of the repressive histone modifications like K27 trimethylation, also the methylases like EZH2, the polycomb proteins can stick and potentially be transmitted to multiple generations.
You mentioned hereditary transmission of some of these epigenetic changes. I know Craig Mello showed some years ago, his group showed some years ago that microRNA, I think it was LET7, could be transmitted by the male mice that had been starved, and the subsequent generations were insulin resistant. So fascinating. Lily, you want to ask the next question?
Okay. So the next question is, how long could an epigenetic effect last? In the case of glycemic control, why wouldn't change of environment induce another epigenetic effect that would reverse beneficial effects?
That's a great question. I wish we had the cohorts to study. This is a question which I always ask, but as far as the DCCT, we don't have the capability to do that because that study can never be repeated. But in collaboration with Sarah Shuck in my department, we are fortunate to start getting samples from the TEDDY clinical trial. The TEDDY is a continuously running, ongoing prospective clinical trial where they're collecting samples from type 1 diabetes, pre-diabetes. So this is going to be a long-term clinical trial from the same subjects. They're going to keep repeatedly collecting samples. So this will tell us whether epigenetic changes starts before the appearance of autoantibodies, during the development of type 1 diabetes, after the appearance of hyperglycemia, how long it lasts. So this study will last well beyond the time that I'm even alive. So I expect we will get these kind of answers. And a lot of people are now getting samples from TEDDY. There's also something called type 1 diabetes exchange. And the type 2 diabetes people are also following up patients. Now you know that this can, depending upon the time of collection, it's very important, the time of collection and the time of follow-up. We can do this of course, in mice, and it's much easier to do that. So we are doing some kinds of studies to answer this question.
Rama, just fascinating, metabolic memory. If you don't mind, we'll have a presentation from Li Lai. She has some work to follow on from yours. And while she's setting up, I just want to remind everyone that you can join by text. Text DEBAKEY to 37607 and text in your message, or join us by web. Go to pollev.com and enter DEBAKEY for questions. So why don't you go ahead with your presentation then, Lily.
Thank you.
Hello everyone. So my topic is about the cardiovascular regeneration through transdifferentiation, where the fibroblast cells can be reprogrammed to become endothelial cells, and how metabolism and epigenetics couple together to regulate this process. So our lab previously found that innate immune signaling activation is necessary for global changes in the expression of epigenetic modifiers to enhance chromatin remodeling and nuclear reprogramming. And we further discovered that cell autonomous innate immune signaling is required for transdifferentiation from fibroblast cells to endothelial cells. So based on this finding, our lab developed a small molecule-based in vitro protocol for the androgenic transdifferentiation from fibroblast cells to endothelial cells. So in this protocol, we firstly use poly-IC, which is agonist of toll-like receptor 3, and then we add endothelial growth factors to the media. So after 2 weeks of treatment, we get a yield of 2 to 3% of fibroblast cells transdifferentiating into endothelial cells. So we call these induced endothelial cells IECs. So the IEC obtained from this protocol manifests high fidelity for endothelial cell lineage. They incorporate acylated LDL, 4-tubular network, have similar transcription profile with genuine endothelial cells, and increase capillary density and perfusion when injected into the ischemic highland of the mouse. So more recently, to test the clinical relevance of this phenomenon, our colleague Xu used the lineage tracing approach to study the transdifferentiation in vivo. So she firstly generated the FSP YFP mice, where all the fibroblast cells and their progenitors that ever expressed fibroblast cell-specific protein FSP1 will be labeled with YFP. Then she performed the ischemic highland mouse model on those lineage tracing mice. And then she observed a significant contribution of the FSP1 originated cells in the CD144 expression endothelial cells during the recovery of the surgery. And this work provided the first evidence of the transdifferentiation phenomenon in vivo in this model. So to further study this phenomenon and enhance transdifferentiation efficiency, I extended the view into the metabolism, which is known to be involved in cell plasticity, and find during this process there is a significant metabolic shift from oxidative phosphorylation to glycolysis, which is known as Warburg effect. So by using widely accepted inhibitors of metabolic enzymes for glycolysis regulation, I further observed that the stimulators of the glycolysis increase the IEC yield, while the inhibitors of the glycolysis decrease the yield of IEC by using our in vitro model and the matrigel plug in vivo model. So those data proves that this glycolytic switch is required for the transdifferentiation in vitro and in vivo. Also, it is known that metabolism pathway activation are not only for the energy supply, but they also generate numerous metabolic intermediates to serve as substrate for epigenetic regulation. So by U13C glucose labeled targeted metabolomics, I found a citrate accumulation during transdifferentiation, which is after toll-like receptor activation by poly-IC. The interesting about citrate is it can be converted to acetyl-CoA, which plays a significant role in epigenetic regulation. So the following experiment proves that the acetyl-CoA increased and the histone acetylation transferase activity increased after poly-IC. And more importantly, the histone acetylation is enhanced. So, and this metabolic event is coupled. So to summarize my work is the transdifferentiation of fibroblast cells to endothelial cells is accompanied with a glycolytic switch, and this metabolic switch is necessary for transdifferentiation in vitro and in vivo. And this metabolic event is coupled with epigenetic regulation by activate citrate ACL pathway to further open chromatin configuration and ultimately enhance transdifferentiation in vivo and in vitro and the vascular regeneration. So now we are testing this concept in vivo by using fibroblast cell-specific conditioned knockout mice. I think it would be very interesting to see how the diabetic condition would affect this process and how to make adjustment of our approach accordingly. Yes.
Thank you, Lily. So Rama, this is work that is a continuation of what we've been doing for a few years now, showing that innate immune activation, stimulation of pattern recognition receptors on cells like fibroblasts, for example, cause epigenetic alterations, global epigenetic alterations. So inflammation causes alteration in the HATs, the histone acetyltransferases, upregulating the histone acetyltransferases, downregulating HDAC. So changes, global changes in epigenetic modifiers that throw the chromatin into an open position. So inflammation opens up the chromatin, which is what you were talking about much earlier in your talk. Furthermore, we've shown that iNOS translocates to the nucleus and starts nitrosylating epigenetic modifiers to change their behavior. So, for example, the PRC complex, which is a repressive complex, it gets nitrosylated and falls off the chromatin during inflammation. Another way in which the chromatin gets opened up. And then Lily has found a third mechanism by which the chromatin becomes opened up, and that this is this glycolytic switch that occurs during acute inflammation that supplies more citrate to the nucleus, allowing for acetyl-CoA metabolism synthesis and histone acetylation. So what do you think about this idea that inflammation can trigger an open chromatin configuration? Oh, she's on, you're muted. Sorry, Rama.
Okay,
there
sorry
you go.
about that. Thank you, Lily, for your fascinating talk. I think this is a great direction that you're taking, because many people have looked at transdifferentiation, but from endothelial, endo-MT, but you're looking at fibroblasts to endothelial cells. I think that really has translational implications too, because can you hear me?
Yes.
Okay.
Yes, we can.
Yeah. All right. Because I couldn't see anyone, so I thought I lost you guys. And you're showing that you can actually improve the hind limb ischemia, improve the function in the hind limb ischemia model. So, have you looked at the fact that you found epigenetic changes is extremely interesting, and what John was saying, that the iNOS regulation in the nucleus and the acetylation of the affecting the PRC complex, all that is very interesting. My question was, is this specific to fibroblast cells, or are there any other cells that can be converted to endothelial cells? You know, like macrophages, can they also be transdifferentiated under the same condition to endothelial cells?
Go ahead, Lily. You want to answer that question?
I think there are other cell sources for endothelial cells, which people are using as transdifferentiation. So. The
answer to your questions, we don't know, but I suspect that this is a phenomenon that is widespread. This what we call transflammation. It's basically inflammatory signaling opens up the chromatin, and we're looking at the ways in which that happens. So acutely, a cell, when it's confronted by a cellular challenge, when a cell is confronted by damage or a pathogen, activation of pattern recognition receptors on that cell will trigger inflammatory signaling, which is well known, well described. And those same pathways, what we found, which is what is new, is those same pathways cause global changes in epigenetic modifiers that open up the chromatin. That's what we found. And we're working through the mechanisms right now. The work started back in about 10 years ago, when we found that inducing a fibroblast to a pluripotent stem cell required this inflammatory signaling. Without it, you could not get an iPSC.
I remember, your seminal work in that area. So I think this all makes sense. And if you, just like fibroblasts can be converted to endothelial cells, can be converted to other cell types too, depending upon how you tweak them, right? But the fact that you found it's just with the poly-IC, you can do it. It's very interesting. In vivo, can you cure impaired angiogenesis in a model of diabetic retinopathy or some of that?
That's a great question. We're looking into it, seeing if we can manipulate this process, this transformation process, to enhance angiogenesis. But what I can tell you is that, as Lily mentioned, we published a paper last year in Circulation. Xu Meng was the first author, showing that during ischemia, there's a subset of fibroblasts that actually can transdifferentiate into endothelial cells. So we have that data by lineage tracing and by single cell analysis.
That was my next question, yeah.
Yeah, there are subsets. We found 8 subsets by transcriptional profiling of fibroblasts, 2 of which seem to participate in angiogenesis. One cluster by making angiogenic cytokines, and another cluster by expressing endothelial function genes, and they seem to be able to transdifferentiate into endothelial cells. We actually found surface markers for these fibroblasts, and which allowed us to sort them out from the hind limb and study them in vitro. And this one set of fibroblasts was able to become very endothelial-like in their expression of surface molecules and formation of tubes in matrigel. And Lily, maybe you can answer this question. Does the fibroblast transform in this process? Does it transform morphologically into an endothelial cell? What have you seen?
So the IEC that we generated, we have been evaluate them in different layers. So we do a lot of in vitro experiments, including tubular formation, which is a standard endothelial cell assay, and we see these IECs they form tubular network on the matrigel, and also when we inject these cells into mix with matrigel and inject these cells with matrigel into the skin mice, we can also see the network formation.
Blood vessel.
So, but however, if it's 100% genuine endothelial cells, we still have to do more evaluation because endothelial cells has so many different types. So so for next step, we also need to work towards to generate specific endothelial cells by using our approach. That's our goal.
Well, we got another question from the audience, actually. I direct it to you because it's really interesting, and I think we can talk about this for the last few minutes. Can epigenetic modifications, such as those you've described to us today, can they play an intermediary role in a process that leads to more lasting effects? So can you get, I think the question is here, can epigenetic alterations lead ultimately to genetic changes that are more persistent?
Epigenetic modification cannot be used to alter genetics. So you actually have to use CRISPR to change the DNA sequence. But you can alter regulatory regions to alter the expression of genes. So you can alter the epigenetic changes around promoters and enhancers. So that's the beauty of epigenetics. Without causing long-lasting genetic damage, you can actually alter a function of a gene. So if a gene is not expressed sufficiently, you can increase the expression of the gene by adding more epigenetic activating marks to the promoter. So the advantage of epigenetic editing is like a non-toxic way of doing it. Because most people are afraid to change your genetics, right? Because you don't know if you fiddle around with your somatic genetic, how it's going to interfere. Because there's also long-range interactions where you have, the chromatin is looping, and if you fiddle around and you change the gene sequence of a particular gene, it can affect other nearby genes too. So on the other hand, if you alter the epigenetics around a gene to make it less expressed, more expressed, or silence it, by adding DNA methylation or a repressive polychrome mark, it is less toxic. And the question is, can it last for a long time? Yes, sure. But DNA methylation, I was just telling you that in stem cells, it can last through multiple generations. So it hasn't been tested in humans. We know that clinical trials are already on now for CRISPR-Cas9. Look for hyperlipidemia. There are companies now doing CRISPR-Cas9 for PCSK9 and also maybe LDL receptor. But to do epigenetic engineering, what I was told is these kind of clinical trials are just starting right now.
That's fascinating. It does seem like a less toxic, genotoxic approach right?
Yeah. I mean, clearly for a genetic disease like sickle cell or, what the other one, cystic fibrosis, those kind of, you need to alter a single gene. But in the case of diabetes, it's a polygenic disease.
Right.
No two people have the same genetic alteration. Cancer is easy to tackle if they are single mutations, just a RAS mutation or a particular mutation is codes for specific types of lung cancer. At City of Hope, they have a very big precision medicine program where nowadays, the patient comes in immediately, his genes are sequenced, and within 2 days, they know exactly what mutation he has, and they are treated exactly with that particular mutation. Our dream is we can do this for cardiovascular disease. My son is a population geneticist, and that's exactly what he does. So he's all the time looking at the particular genes coding for hyperlipidemia, and they treat accordingly. It's a little bit more difficult for diabetic vascular complications.
Mm-hmm. One last question. I think it's an important question from the audience because the extracellular matrix does become altered in diabetes. The question is related to what is the role of the extracellular matrix in epigenetic modifications? I suppose it could be like outside-in signaling, right?
Mm-hmm. Yes. We definitely know, and extracellular matrix is something that is this is something we've been working on for a long time, integrins and fibronectin, collagen. We actually showed that collagen, we published the first microRNA in the kidney where we were fascinated to see that it regulates collagen extracellular matrix deposition in the kidney, and also we have data in the cardiovascular system. So we know that the extracellular matrix actually dictates the behavior of a cell. So clearly, when the extracellular matrix is altered, the signal transduction mechanisms that are altered, through the beta-1 integrins or the other alpha integrins, this signal can alter the particular transcription factors that collaborate with the epigenetic machinery. So they're all tied in together.
Well, Dr. Natarajan, I think we're coming to the close of our program. It was really a pleasure to have you. We learned a lot about metabolic memory today and epigenetic alterations that play a role in cardiovascular disease. So thank you so much for joining us.
Thank you, and thank you, Li Lai, for your great talk. Thank you, John. It's very nice to catch up after so many years. Yeah. Li Lai, you might be interested that I used to follow John's work. We were all postdocs in those good old days. I used to follow all his fascinating work on eNOS and nitric oxide. So we go a long way. Anyway.
Thank you, Rama. Bye-bye now.
Bye.
And thank you all for joining us today at the cutting edge of cardiovascular sciences.